My name is Andrew Binley. I'm based at Lancaster University in the UK. I'm going to give a talk here on electrical resistivity imaging of denitrifying permeable reactive barriers. This is work that's led primarily by Lee Burberry at the Institute of Environmental Science and Research in New Zealand, and also supported by Theo Saris, Richard Mellis, Mike Finnemore, Giorgio Cassiani, and Michael So. What we've been doing is carrying out a number of electrical resistivity imaging experiments alongside tracer tests to look at the hydraulic properties within and around a test permeable reactive barrier setup at a site in New Zealand. Many parts of the world are experiencing increasing concentration of nitrate in groundwater. This so-called legacy nitrate is a result of historic agricultural activities. And although some countries are taking steps by changing the way we manage the land, for example, reducing nitrate fertilizer application, we still need a way to be able to handle the current elevated nitrate concentrations in some groundwater bodies. Permeable reactive barriers, PRBs, may be one tool that we can use to address this. PRBs are engineered barriers, commonly consisting of zero-valent iron for the treatment of chlorinated solvents. But other materials are used for the treatment of nitrate in groundwater. Solid carbon is used within the trenches to intercept the nitrate contaminated groundwater flow. And then some denitrification takes place within the barrier, thus reducing the nitrate concentration that flows through the barrier. Wood chip denitrifying PRBs have been used in a number of countries. A particular challenge of their use in the area that we're looking at here in the Canterbury Plains of New Zealand is the high heterogeneity and high permeability of the aquifers. So we have very high advective groundwater velocity, and this makes it particularly challenging given that we actually need a barrier to be even more permeable to ensure that groundwater flows through the barrier and not around it. And we also need to think about the resulting residence time of groundwater within the barrier to ensure that there is sufficient time for reactions to take place. Our study site is in the Canterbury region of New Zealand. And in this study site, we've been looking at two different types of barrier materials and assessing the performance of these barriers for reducing nitrate concentration. So this is an overview of the site plan. We've got a number of wells. We've got groundwater flow from the left to the right. As you can see from these equipotentials, we've got a barrier location here with two cells shown by in this insert, and we've got a number of wells within the barrier and around the perimeter of the barrier, allowing us to monitor the hydraulic conditions but also the nitrate concentrations within the formation. From pumping tests carried out at the site, we estimate the hydraulic conductivity of the typically 5 meter thick gravel aquifer to be over 1,000 meters a day. So it's a very permeable system. We've got high advective velocities, and we've also got high heterogeneity within this gravel system. And we've been using this site for carrying out a number of tracer tests to look at flows through the aquifer and also flows through and around the barrier. So the two cells being studied here are shown here. We've got one in cell 1, which consists of a mixture of gravel and chipped wood. And in cell 2, we've got hogged wood, which is splintered, crudely broken up wood. We estimate the hydraulic conductivity in the cell materials to be 20 to 30,000 meters a day. So some 20 to 30 times what we estimate the aquifer permeability to be. These estimates of hydraulic conductivity are on after installation of the barrier, but we need to think about how this changes over time. So does that lead to a degradation of the barrier? The entire 2-cell system is 25 meters long by 5 meters in plan, and it's 3 meters deep, so penetrating part of the gravel aquifer. After installation in 2018-19, sorry, if we look at before and after installation of the barrier, we can see here nitrate concentrations. So the red indicates high levels of nitrate, relatively high levels of nitrate. And then after the barrier was installed, we can see a change in the concentrations down gradient of the barrier, suggesting that there is some effectiveness of the 2 cells in reducing nitrate concentration. But the question is, does that remain effective over time? Is there some change to the effectiveness of the barrier? For example, some blockage of the barrier material, or some reduction in its chemical effectiveness over time. So a number of experiments have been conducted at the site to study this, and I'm going to focus here on how we've been using geophysics to assess this. And we've been using primarily electrical resistivity tomography, ERT, to track saline traces injected up gradient and also adjacent to the barrier to look at the movement of salt tracer through the barrier and through the aquifer, and get some idea on its hydraulic status, on the barrier's hydraulic status within the aquifer. We've also been using some flow and solute transport models to in combination with the geophysical data to try and interpret our results. We've carried out a number of tracer tests, and I'm going to show some examples of some here. Our first tracer test started in June 2020. We had an injection C4, which is just shown here, which is up gradient of the 2 cells. We used an instrument that was a little bit limited in terms of the data collection speed and so on. This is our first trial. And then after that, in September 2020, we did another test, which was in injection in a well away from the barrier. And then we moved to a different instrument, the ZZ Geo system, which allowed more 3D type measurements to be collected, much higher frequency of data collection. In December 2020, we then moved to another tracer test near the cells, and then in 2021, February 2021, we had another test at N4, which is seen to the north of the cells, and then 2 further tests, April 21 and May 21. And I'm going to show a collection of some of the data that we've collected from these experiments and some of the interpretations. We've modeled the data using the code R3T, so it's a three-dimensional unstructured mesh. We know where the barrier is. We can fit that within the mesh, and then we typically have 3 or 4 lines of electrodes. We use 3 lines for the first experiment, 4 lines for the other experiment. And as I said before, the first experiment was limited in terms of the types of number of measurements that we could take because of the instrument. So they were kind of more like 2D, quasi 3D measurements for the subsequent tracer tests. So we used true 3D type measurements within our data collection scheme. So let's look at the first tracer test. We've got 3 lines of electrodes here, an injection well C4, and what should happen is the tracer should go in here, and then we're expecting the tracer to move through. Maybe it passes through cell 1, or maybe it passes through cell 2, or maybe it goes through both of these. And so we can come up with an estimate of what we think the travel time should be based on the dilution of the tracer at injection, and that allows us to design our or adapt our data collection time for the experiment. Here is a section of the ERT result prior to injection. So C4 is the injection point, and we will see the low resistivity, the blue zone as the barrier, and the red zone is the gravel aquifer adjacent to the barrier. Now, if we look at the tracer test results here, showing this different parts of the image from the same tracer test, we'll see the progression of this. And here we're looking at differences. So this is a difference inversion, and blue color represents an increase in conductivity due to the saline tracer. And what stands out here is that we see the tracer moving towards the barrier and then seems to move laterally along the face of the barrier, suggesting that it's actually not passing through the barrier, but is actually running outside or underneath the barrier. And this is supported by our observations of minimal capture of saline water in our monitored borehole C5, just down gradient of the barrier. So this was through us because we really expected to see the increase in conductivity, electrical conductivity, focused through the barrier. We did some solute transport modeling using the code CWAT, calibrated with PEST, and we set this up with a parameterized barrier separate to the aquifer, and then we tried to match the model against the observation wells. And if we do that, we see this kind of plume evolution. Whereas if we try to use the ERT data along with the observation wells, then we see a completely different behavior that suggests that the solute is not passing through the barrier, or most of it is not passing through the barrier, but is actually passing around the barrier. So we get contrasting behavior there, whether we use the geophysical data or not. We then ran a tracer test away from the barrier, injecting an SS4, shown here. And we use this, first of all, to test the new instrument that we're using for more high frequency data collection, and in a 3D configuration. So this is our pre-tracer image of resistivity, and we injected the tracer SS4, and we looked at the evolution of that tracer with the difference ERT images. And we'll see now the sequence of this as it moves through the system. And we can see here that the plume moves as expected along the groundwater, the ambient groundwater flow path that we know exists at the site. So this was reasonably well behaved tracer experiment, nice and simple, and behaved as expected. So the SS4 tracer moved as expected, but why didn't we see that same kind of behavior with this C4 injection at the PRB? So we went back and ran a number of other tracer tests, and in this case, at injection at M4, which is slightly south of the, or just south of the barrier. And we were looking here to see whether the tracer would actually move into the barrier, as it should do, given that this should be a high permeable feature, or whether it actually moves around the barrier. So here is our pre-tracer image showing the blue zone, the high conductivity, low resistivity, due to the nature of the material in the barrier. This is our expected flow direction. And then if we look at a sequence of images, we can see now that the tracer does indeed seem flow around the barrier and not towards the barrier, which is what it should do, given that this is should be a high permeability unit within the aquifer. And we can compare our results with observation wells made in some wells just outside and within the barrier. And we can see here from this profile, M4 is the injection. So this is the decay. The black line is the decay of the tracer. And then if we look at PX2, which is just adjacent to the barrier, just up gradient of it, this is the green line. So this is the tracer moving through there. And then if we look at S5 over here, which is away from where it should be going, given that it should be moving towards the barrier, but S5 shows a similar behavior, which is kind of supporting our ERT observations. And PX4 within the barrier shows a much more delayed response. So we're seeing some movement through the barrier, but not as much as that's moving away from the barrier. And these are all suggesting that the barrier is in fact not performing as designed. Another experiment I'm going to show you here is now an injection at N4, which is on the north side of the site, again upgrading of the barrier, pre-tracer ERT image showing the blue zone where the barrier is, the red zone where the gravel aquifer is, and now we look at the injection at N4, and indeed, as before in the M4 experiment, we're seeing the tracer tending to move around the barrier and not through the barrier. And these data are supported by our observation wells, where we measure locally the electrical conductivity of the tracer. So in summary, we've shown the results from a series of salt tracer experiments using time-lapse ERT to monitor the flow and solute transport pathways within and around the barrier. And these have revealed some really complex characteristics of the solute pathways. And some of the results suggest that the PRB is not really performing as well hydraulically as designed. And these have been supported by independent observations in monitoring wells. Some very recent invasive investigations of the barrier materials suggest that clogging has not occurred. So we believe that these complex flow pathways are actually not a result of the barrier, but a result of the heterogeneity of the host aquifer, particularly down gradient of the barrier. We're currently exploring how to combine ERT and solute transport modeling, i.e. coupled hydrogeophysical inversion, to characterize the hydraulic properties of the barriers and the aquifer. And in addition, we're planning some DNA tracer injections at multiple locations at the site to try to differentiate flow pathways, and we'll be comparing those with time-lapse ERT.